The overall goal of this experimental method is to determine the function of high-density lipoprotein using a biochemical cell-free assay that measures the lipid peroxide content of a specific amount of HDL.
This method can help answer key questions in several fields of medicine, such as the role of HDL function in cardiovascular disease and metabolic abnormalities. The main advantage of this technique is that it is a cell-free biochemical method that can be used to reliably determine HDL function.
Though this method can provide insight into the role of HDL function in cardiovascular disease and metabolic abnormalities, it can also be applied to other studies of disease, such as inflammatory diseases.
To begin this procedure, prepare the working layout for the 96-well plate, including the appropriate controls, samples, and replicates as outlined in the text protocol. Calculate the volume of reagents needed based on the number of samples and replicates. Using a permanent marker, label tubes for each step in the assay. Next, combine 10 microliters from each sample to create a pooled control sample. Create a second pooled control sample for the clinical laboratory in order to determine the HDL-C value. After preparing a large stock of HDL, cryopreserve several aliquots to minimize freeze-thaw cycles. Pool at least 10 different replicates to create a pooled quality control. Then determine the average value of HDL-ox as outlined in the text protocol.